Everyone, so welcome to our new lecture. In this lecture, we are going to study chapter number 8, that is useful and harmful microbes. In the previous lecture, we have studied about one of the useful microorganism, and that is Lactobacilli. So we know that Lactobacilli, it is a blue colored rod shaped bacteria which is widely used in the production of foodstuffs. Okay, so we have studied thoroughly about the application of Lactobacilli in our previous lecture. Now, in this lecture, we are going to study one more useful microorganism, and that is Rhizobium, or also called as Rhizobia. Okay. Now, as Lactobacilli was a bacteria, similarly, Rhizobium is also a bacteria, but it is categorized as symbiotic bacteria. Okay. Now, let us see where do we find this particular bacteria. Okay, we have studied that lactobacilli is found, or we can see lactobacilli in a milk or milk products. Now, rhizobium or rhizobia can be observed, or it can be seen in a plants which are leguminous in nature. Okay, now to understand the nature or the behavior of this particular bacteria or the role of this bacteria, we can perform or conduct a simple experiment. Now, what is the experiment? Let us take a plantlet of soya bean. Okay, so we are going to take plantlet, okay, one of the part from the root of soya bean plant. Okay, we will sterilize it in 3 is to 5% of hydrogen peroxide. Okay, we will take the plantlet of soya bean plant, okay, and then we will sterilize it, and we are going to use the solution that is 3 to 5% solution of hydrogen peroxide. Okay, now we will take that solution and the soya bean, and we will keep it in 70% of ethyl alcohol for 4 to 5 minutes. Okay, so after sterilizing the plantlet of soya bean, we will again keep that soya bean plantlet, okay, in 70% of ethyl alcohol, okay, for 4 to 5 minutes. So ethyl alcohol, it is a compound, okay, that is C2H5OH. Okay, what is the formula of ethyl alcohol? The formula of ethyl alcohol is C2H5OH. Okay, this is the formula of ethyl alcohol. Now, after this, the another step is take thin nodules of root after sterilizing it and place it in the safranin solution for 2 to 3 minutes. Okay, so after keeping the soya bean plantlet, we will take out the root, okay, very thin nodules of those root, okay, very thin part or structure from that plantlet. We will sterilize it again, and we will place that nodule in a safranin solution, okay, and the duration will be 2 to 3 minutes. After performing all these steps, we can observe that specimen under a microscope. Okay, so after doing all these 3 to 4 steps, we will get a specimen, and that specimen will be placed on a cover slip. Okay, on a slide, and we will place the cover slip, and then we will observe that specimen under a microscope. What will be the observation? The observations are going to be, we can see the pinkish rod shaped organisms. Those are nothing but rhizobium bacteria. Okay, so our first initial observation will be that we will see some organisms which are rod shaped, okay, which will have a shape like a rod. The color of those organisms is going to be pink in color. Okay, so those organisms are nothing but rhizobium bacteria. Okay, then they are found in root nodules of leguminous plants. We have to keep this in mind, that rhizobium bacteria are found in root nodules of leguminous plants. So the plants which are leguminous in nature, we can find this bacteria in those plants only. Okay, so as we can see, this is the nodule, root nodule, and in this root nodule you can find the rhizobium bacteria. Okay, now this is the microscopic image of the bacteria. Okay, this is the microscopic image of rhizobium. Now let's see the roles and importance of these bacteria. So what are the roles and importance of rhizobium? They are very much useful in the supplies of nitrates, nitrites and amino acids to plant. In exchange gets energy in the form of carbohydrates, that is symbiosis. This process is called as symbiosis. So what is the main role of rhizobium bacteria? Okay, this bacteria helps to provide some important nutrients to plant. Okay, so for the good growth of plants require some nitrate molecules. Okay, it requires some nitrites, and amino acid is also necessary for the good growth of a plant. Okay, and all these important compounds, nitrates, nitrites and amino acid, are provided to the plant by this bacteria, which is called as rhizobium. Okay, now definitely it is working. Rhizobium is performing some work, and to perform that work, rhizobium requires some energy. As rhizobium is a living organism, definitely it will require some energy to perform that work. Okay, to supply the nitrates, nitrites and amino acid to the plant, rhizobium needs some energy to do that. And this energy, rhizobium has this energy from the carbohydrates of the plant. Okay, so whatever carbohydrates which is present in the root nodules, rhizobium is going to extract those carbohydrates to gain the energy to perform this work. So this is the good relationship between the plant and the bacteria. And such relationship between plant and bacteria, or such coordination between plant and bacteria, is called as symbiosis. What it is called? It is called as symbiosis. Okay, now another role, nitrogen fixation of leguminous plants are done by rhizobia. So leguminous plants like sweet pea, soya bean, okay, all these leguminous plants needs nitrogen for their growth, and this nitrogen fixation in the soil is done by the rhizobia bacteria. Okay, due to this, beans and pulses are rich in proteins. Now as the nitrogen fixation has taken place, okay, and the plant is growing in a very good way, definitely those plants are going to be rich in proteins. Okay, so the quality of the plant is increased by these bacteria called as rhizobium. Now after harvesting crops, their leftover roots and other parts are dumped in the farm soil to maintain the bacterial population. Now after harvesting all these beans and pulses, okay, or cereals, what farmer can do? The farmer can do simple thing, that definitely we harvest the upper part or the required food material from the plant. Okay, and then what we do with the roots which are not required to us, we throw it away or we burn it away. But that is not the good way. What else we can do? We can dump those root inside the soil, okay, farm soil. And by doing this, what we can ensure? We can ensure the bacterial population. Okay, so rhizobium bacteria will sustain in that soil, and definitely when we will plant new crops, definitely they are going to be benefited by that rhizobium bacteria. Okay, now if we do this, what we can achieve? We can achieve the less use of chemical fertilizers. Okay, we will eradicate the use of chemical fertilizers. We use chemical fertilizers to improve the yield of the crop. Okay, instead we can use all these biological methods, okay, to maintain the bacterial population, to increase the quality of the food product. Okay, and definitely these methods, biological methods, are eco-friendly in nature. Okay, so that will ensure the good environmental condition. So these are the roles and importance of rhizobium bacteria. Now let us see what is rhizobial inoculation. So seeds are coated with rhizobial solution or powdered before sowing. After sowing, rhizobia enter the plantlets, helps in the supply of nitrogen to cereal and other crops besides leguminous crops. Now what we have seen generally, that rhizobium bacteria is found, or it is found in leguminous crops. Okay, but what about the other types of crops which are not leguminous in nature? Definitely they lack this bacteria which is called as rhizobium. Okay, so if we want to increase the quality of other food crops, what we can do? We can coat the seeds of all other food crops by a rhizobium solution. Okay, rhizobial solution or powder. And this will ensure the good nitrogen fixation for those crops also. Okay, so this process is nothing but rhizobial inoculation. Okay, so this is all about the rhizobium bacteria and the application of it. So in this lecture we have seen about rhizobium bacteria. For this lecture, your homework will be you have to write down roles of rhizobia in your notebook. In the next lecture, we will study about one more useful microorganism, and that is yeast. Okay, so we will meet in the next lecture. Until then, goodbye.